We will now be demonstrating endocytosis. Endocytosis is the process by which cells take in bulk particles that are too big to just flow through the membrane. So let's say we have a food particle that's too large. A vesicle is formed in the cell membrane. As the vesicle is formed, it flows back into the interior of the cell, into the cytoplasm toward the nucleus. The membrane then continues and reforms, and the process in terms of engulfing that or taking that particle in is completed. Now, receptor-mediated endocytosis is specific to a particular molecule. If we use the same format and use the same bulk particle, and that bulk particle tries to enter the cell, but doesn't have the right initiators, it won't fit. So now reject that back out, so it won't take, it doesn't catch. Why? Because it doesn't have the right receptors. But let's say, for example, if there's a HP receptor on the cell, and we have an HP computer or HP bulk particle, that HP bulk particle is accepted into the cell because it fits. Then the particle rolls, the vesicle rolls into the nucleus of the cell or into the cytoplasm only because it's a specific receptor on there, and that is receptor-mediated endocytosis. This is phagocytosis. If we have our team that's demonstrating a white blood cell and we have the chair that is a bacteria or virus, when a virus or bacteria enters the bloodstream or a cell, it disperses what's called a chemoattractant, which activates our white blood cell. Once it's activated, the white blood cell hunts down, finds the bacteria, and engulfs it, takes it into the interior of that phagocyte, where the lysosomes break that thing down and get rid of it out of your body. Right now we're talking about penocytosis. And what penocytosis is basically the way that a cell drinks. Sometimes in the proteins of the cell can actually take in water molecules individually through those proteins. Other times when the water is too big to take in through those proteins, the cell has to do what's called penocytosis. And that's when the cell creates a vesicle in the phospholipid bilayer and engulfs those water molecules, taking the water molecules from outside of the cell, engulfing them, and separating itself and bringing it inside the cell, where those water molecules are then digested by the lysosomes in the cytoplasm of the cell. In this video, we will try to display active transport. In active transport is when a molecule wants to go from an area of low concentration inside the cell, in this case, to an area of high concentration, which is outside the cell. But to do that, you need some energy. So we're going to use this coffee cup as ATP. So our molecule will take in some ATP, and now our molecule has enough energy to force itself outside of the cell into the crowded area. And now the concentration gradient has changed by way of using ATP. Thank you. When we try to explain passive transport, if we look at this room as a cell and the doorway is a transport protein. So we have Jasmine and Jayla. They are molecules inside the cell, and Tamia is the molecule outside the cell doing their thing. But the concentration gradient inside the cell is greater than it is outside. So the molecules will move along the concentration gradient to get from an area of high concentration into an area of low concentration. At this point, the concentration gradient has changed, and they still want to get from high to low. So now Tamia comes in and doing her molecule thing, and now the gradient has changed once again. So we have another molecule that moves outside using the passive transport protein that is our door, and this is the process of passive transport inside the cell.
Action.
Who is it?
It's your primary messenger.
The receptor protein always got to answer the door. What you want?
Got a message for you. You're welcome.
Thank you. What we gonna do about this? I don't know. What is this? This must be for the dental cycle. So what we gonna do? I don't know. I'm about to give it to them. Deliver the message. Okay. Cyclase AMP.
Cyclic AMP, we can do that. Thank you.
This picture is showing enzymatic activities. These are two enzymes. This is cell 1. This is cell 2. Cell 2 have different subs that are sending to the enzymes, and they're sending to the enzymes to get a response. But these are two different enzymes, and they only fit certain enzymes, and they have to be a perfect match in order to get a perfect result.
Recognition, and these are the phospholipids that's surrounding the cells. Actually, the glycoprotein is trying to recognize the cell, which the binding is short-lived, and the glycoprotein cannot bind with any other cell, which we go into intercellular protein that these cells may hook together to the junctions, and the binding is long lasting, and that's it.
Okay, so here we have our cell. Here we have the ECM. Then we have our fibronectin, which is in blue. Then we have our phospholipid bilayers. This is our protein, and inside the cell we have our cytoskeleton.
So what we'll be explaining is kind of how the ECM and the cytoskeleton connect using proteins to stabilize the cell. You have your extracellular matrix on the outside, and fibronectin, which comes from inside the cell and actually connects with the proteins on the cell as well. So here you have your extracellular matrix, fibronectin, which is made out of proteins, and you have your protein here embedded in the bilayer. The protein connects and binds here with the cytoskeleton, and what that does is that stabilizes the cell on the inside and outside to help it maintain its shape and function.
This is the light compound microscope. This is a picture of how it looks. The compound light microscope has its own light source. It forms images of small objects consist of object lens with very short focal lengths, and both lens mount in the same tube. And the picture kind of show the eyepiece, the final imagine, where the eye goes, the project lens, the objective. This is the specimen, this is the contact lens, and then the light at the bottom.
I'll be discussing the scanning electron microscope, which is a microscope that produces images of a sample by scanning the surface with a beam of electrons. The electrons interact with atoms in the sample, producing various signals that have info about the sample surfaces and composition. The scanning electron microscope is high resolution, 3-dimensional images, and it requires a stable power supply. This is my drawing of the scanning electron microscope. It has an electron gun, condensed lenses. It has a sample chamber with an X-ray detector.
This is the transmission electron microscope. Part of it. This is how it looks. This is the electron. This is the electron source. This is the electron beam, and this is the lens that you see out of, and this is the viewing scan. Then I'm going to take you over to what it do, the technique used to be observed, to the features of very small specimens. And the gun fires the beam of the electron, as you can see in the microscope. And that's transmission electron microscope.